A very warm and happy morning to our speaker of the session, Dr. Lakshmanan Jagannathan, and all teachers and my fellow students. It's a pleasure to welcome you all to this virtual webinar on drug discovery and development, current technologies and career opportunities, organized as a part of a Silver Webinar series. Prayer is intimacy with God that leads to fulfillment of His purposes. Now, we will start this event by seeking the blessings of God. I request Executive Committee member of Neuberg Group, Ms. Jayalakshmi from second year BSc Biochemistry to present the prayer song.
Vyavrutham satyaparam trusatyam satyasya yonam nihitancha satye satyasya satyam ritasatyanetram satyatmakam tvam sharanam prapannaha vani gunanukathane shravanam kathayam hastau cha karmasu manasthava padayonaha स्मृत्यां शिरस्तव निवास जगत्प्रणामे दृष्टिस्तथां दर्शनेस्तु भवत्वनूनां नमो भगवते तस्मै कृष्णायाद्भुतकर्मणे रूपनामविधेयेन जगत्क्रीडति यो यतः.
Leadership is the capacity to transform vision into reality. I take this opportunity to invite our Head of Department, Dr. Srinivasan sir, to address the gathering. Please, sir.
A very warm good morning to all the participants of this program. So our department is celebrating a Silver Jubilee. We are celebrating this year, the entire year, by having a webinar a month. So in that webinar series, today we are having the Silver Jubilee webinar series. Today we have amidst us Dr. Lakshmanan Jagannathan, one of our very known distinguished alumnus of our department. So who is currently working on bioinformatics, that is drug discovery, genomics, transcriptomics, and epigenomic techniques. So we are very fortunate to have him amidst us. So he'll be highlighting us about the various aspects of the drug discovery and the development that is taking place in the current scenario of the research. So I take this opportunity to welcome Dr. Lakshmanan Jagannathan and the participants from the various city colleges, staff members of our department and other departments, staff members from other colleges who are here to participate in this wonderful webinar. So the participants are expected to be enlightened about this particular topic, which is a very hot topic. One of my friend, he is working in US. He is also working on this epigenomic, that is epigenetic, and he is trying to develop a drug for anti-aging process. So I'm also very eager to listen to our the speakers talk today on this. If he touches that epigenomics techniques, it's a very interesting topic. And now the buzzword now during this pandemic era, both in the academic and in research field, is the digital education. So everything is going in silico. So in that perspective, we also have here amidst us the speaker who has specialized in the computational aspects of biochemistry, to be more precise, and all these topics, the omics, we say the omics. So he is an expert in that field. So looking forward to hear his talk on the various aspects of drug discovery and development, and what are the recent trends and the technologies that are associated with the drug development, and what are the career opportunities that are associated with such areas of computational biochemistry, to be very precise to say in that. Or now every area has become a specialized area. The drug discovery itself has become a specialized area, and epigenetic, it's also becoming, as all areas are becoming more and more specialized day by day. So nevertheless, we'll have a overall view or a bird's view of all these areas. I hope that speaker will be touching upon those aspects of the drug discovery and its development, what is happening around the world and various parts of the world, and what are the career opportunities that are available for any prospective student. So I ask the final years, especially the undergraduate and the postgraduate final year students, to pay more attention to the talk and keep all your doubts and questions and reserve those questions. And at the end of the webinar, you can address those questions to the speaker. He will be able to guide you through whatever your aspirations are. So with this short note, I once again welcome all the participants of the webinar for this our Silver Jubilee webinar series. That's the 24th webinar that we have on drug discovery and development, current technologies and career opportunities, delivered by one of our distinguished alumnus, Dr. Lakshmanan Jagannathan. Over to the organizing team. Thank you for giving me this opportunity.
Thank you, sir. Medicine is the science of uncertainty and the art of probability. Now, I request President of Neuberg, Ms. Lakshmi Sri of second year MSc Biochemistry to introduce our speaker of the day.
Good morning to one and all present here. It's been an immense pleasure for me to invite the chief guest for today, who is an alumni of our department on behalf of the Department of Biochemistry, Dr. Lakshmanan Jagannathan, to address the gathering. He has done his UG BSc Biochemistry in DG Vaishnav College in the year 2000 to 2003, and done his post-graduation in Advanced Biochemistry at University of Madras in the year 2003 and 2005. And then he has done his PhD in AUKB Research Center, Anna University, Chennai, in the year 2009 and 2013 with excellence. He started his career in the year 2005 in a research associate in GVK Bioscience Informatic Division. After which he moved as a research associate to literature management and documentation, Vispar Bioscience, PVBA, in the year 2007 to 2008. After which he has done his postdoctoral fellowship in NYU Langone Medical Center in the year November 2013 to February 2016. After which he has worked as a scientist in Vantage Research Private Limited, Chennai in the year 2016 and 2019. And then now he currently working as a Vidant Analyst in Indian Lead Molecular Diagnostic Center, which is called this PerkinElmer Health Science Private Limited. He has a key skills in computational, which is drug discovery, genomics, transcriptomics, epigenomics technique. And he also done many literature review in meta-analysis, data curation, evidence synthesis, network meta-analysis, and statistic analysis. He has many experimental researches in aseptic microbiological technique, biochemical assay, molecular biology, cell biology, and proteomics technique, and epigenetic technique. He has published various publications in many notable national and international journals. Some of them are in association with Kumar R. Tyagarajan K. New L. Marvel K. G. Willis D. Chope B. The research is under understanding IO combination using a clinical OSP model that incorporate heterogeneity in patient response in the year May 2021. And he also attended many conferences in national and international seminars, and he also participated in notable 1R, participated in 10th Cajun Population Approach Group of Indian Workshop in the year 2018. And he also presented a poster titled System Approach to Immuno-Oncology, IO Drug Development, Integrating Data and Knowledge on Indian Society for the Study of Xenobiotic Conference held in ISS Bangalore in December 2018. He's still continuing his education and certification. Some notable certifications received by him are Introductory Human Physiology from Duke University, which he got certificate in the year September 2016. And he also now currently doing all his research work. I would like to continue the session, sir, with your motivating speech. So over to you, sir.
Hello, hi. Thank you very much for the kind introduction. Thank you very much for Professor Srinivasan sir for giving me this opportunity. And of course, the organizing committee and all the members and all the students here. And I'm just really happy to know that there's this kind of webinar activities are going on in our college, and I'm very happy to be part of it. I'll start presenting my topic. So the topic of today's presentation is drug discovery and development, and I'll be talking about the current technologies and career opportunities for the life science students. This whole topic is mostly like from my personal experience, mostly I'm trying to cover it. I know you have been already in some kind of examination or kind of something, so I don't want to bore you out with a lot of information today. Yeah. Outline of today's talk is I would be going through the drug discovery and development process overview, and followed by kind of what is the new trend, new current techniques or um, methods used in the drug discovery and development process. From my personal experience, starting from my PhD to the postdoc and current work as well, I'll be walking you through that information. And I'll also touch upon the kind of what are the career development strategies and tools I personally use to get into the jobs or get into different aspects of my career. So finally, the overall objective of today's talk is to sensitize the upcoming students to the drug discovery and development concepts and provide the direction for the career opportunities. So how the drugs are currently discovered and developed, right? So it is like finding a needle in a haystack, and it's like it takes kind of almost 10 years to develop a single drug, and so if you just take the cost involved in the development of drug, it is almost more than $1 billion used to develop a single drug. And on top of all this thing, the failure rate of drug discovery and development is kind of it's 95%. So in the sense like if you develop 100 drugs, only 5 can go into the market, and rest 100 billion, only 5 billion is can be materialized. Next 100 billion investment is being lost during this process. So that's how difficult is the drug discovery and development currently. So, like I mentioned, I'll be there is a like I just always mentioned. There's a drug discovery and there is a development process, right? So I'll be talking about two different processes here. The first and foremost step involved is like you have one need to understand the disease mechanism. How do we understand this? Is from the various research activities going around different labs, academic labs, or different private industries labs. They always try to understand what are the different disease mechanisms which one need to understand, which one need to if there is any unmet medical needs or anything, they just try to look through all this information and try to understand the disease mechanism. And of course, there will be a lot of molecular studies and gene target validation. All these studies involved. And once we understand the molecular mechanism, finally, like one need to understand, one need to work through the various animal models. We need to select the relevant species as well as the transgenic mouse models were developed. And if there is any chemicals or if there is any inhibitors or anything relevant with that particular process involved, we need to find the relevant resources for the understanding of target selection and validation. Then this all leads to finally, once we understood the molecular mechanism, once we understood the relevant animal model, then we will be like having a confirmed target in our hand, which can be anything, which can be a receptor, can be a transporter proteins, enzymes, signaling molecules. So just to touch upon the value of biochemistry here. I know. I think like we are the groups. We always study all the as in terms of biomolecules in our earlier BSc to MSc. We always read about various receptors, N channels, transport enzymes, everything all over the time, all the time. So that kind of understanding is kind of pivotal. It is vital to have to initiate the drug discovery process. So I feel like having done the biochemistry in my earlier career, I felt that really helped me to understand all the different concepts or different targets involved in the drug discovery process. And so once you understand the target, then now we have to identify the potential leads, which can be a chemical, it can be any agonist, antagonist, any protein, any chemical moiety, which can either inhibit or activate a particular target. So that one needs to identify from the last source from the chemical library or any other resources from the academic research. And once we find the lead is one potential lead is there, then we need to understand, we need to optimize the lead by way of it is increasing its selectivity by studying its tolerability. All this, we need to make sure that the drug, the potential lead, is not causing too much damage or too much activities apart from what is required. So all this information we need to do the experimental analysis and understand optimize the leads. So, for example, if there is one functional group is not working properly, so instead of alpha OH group, if you can change it to aldehyde group, if the function is more better, then we need to change the chemical moiety based on that, and then work on it. Then once we done with all the discovery process, there is you have a potential drug candidate here, and then it needs to be tested in the human studies. There are like phase 1, 2, 3, the clinical trial phase 1 to 3, then finally the molecule is approved. So this is the overall drug approval and registration process. I'll just go through the one aspect after the another, like in detail. Just as I mentioned, like for the target selection and validation, we need to define the unmet medical needs, and then that can be from the academic research, and finally, like then we need to move on to the understanding the molecular mechanism of the disease and identify the therapeutic target, like just I showed you, enzyme receptors, what are the ion channels, and all the aspect of it, and finally demonstrate its target validity using different techniques like RNA technology. We need to knock in into the animal model, and all this all this kind of assays are required to check the validity of the target. Then we move on to the development. The development have stages of preclinical to the clinical. At the preclinical stage, we have to understand the safety assessment, toxicology assessment, drug metabolism. ADME is very critical part. It is like absorption, distribution, metabolism, and excretion. We need to understand those concepts, and then finally, then we need to go through a lot of pharmacological testing, shelf life, and all these things will be tested. There's a bioprocess R&D involved in which the molecules are synthesized in a milligram to gram to kilogram level to check its shelf life and other aspects. Formulations are involved. Then once everything, all the preclinical testing has been done, we have a regulatory affairs process, which is like again a lot of project planning and management, marketing process. All this thing needs to be sorted out. And finally, like once all the data are collected, it needs to be, we need to have a statistical and chronological data collection and the information services like clinical data mining, clinical data research, and all this thing has to go through. And finally, once the drug is, all the information is ready, then it is called investigation drug. Then it needs to be planned for the clinical investigation on the patient side. Now we will move on to the clinical trials part. So what we went through so far is target selection, discovery, and then development in the preclinical. Inside the lab, we have tested the molecule. Right now, then it will move to the clinic. So the drug is now called investigational new drug, and it needs to be there's a FDA application. In India there's a Indian medical Indian drug authority is there, and we need to file the application to get the approval to test it on human subject. So in human subject, the initial part is phase one. It is like in which like we'll be testing it in the 20 to 100 healthy volunteers to check the information about the ADME. So absorption, metabolism, effect on organs and tissues, side effects and dosage. All this information needs to be tested. Once there is a satisfaction in this phase, then it will be taken into the patient population. There should be a comparison between the treatment versus control group and the patients. How the here in this phase we will be checking the effectiveness in the treating the disease, short-term side effects in the health and impaired patients, and what could be the potential dose range we should be applying. All this information needs to be collected in this phase 2 trial. And finally, then the phase 3 trial, which is one of the largest time-consuming as well as the money-consuming process, in which hundreds and thousands of patients will be tested for the given drug, and it takes long time. Even for example, for certain cardiovascular or cancer therapy and all, it takes almost 2 to 3 years minimum to test the molecule in the phase 3. Phase 3 setup. So in this during this phase 3 trial, one would study the benefit risk relationship of the drug. So what is the long-term side effects, and if there is anything labeling that needs to be tested, all this information has been collected and validated during this phase. And there is a phase 4 also, which is like after the approval from the FDA, the drug needs to be continuously monitored for its other potential side effects in the regular market. So I will come back to this particular phase 4 later, because this is where right now a lot of opportunities are there in India. So now the drug has been approved by the process, right? It is not also straightforward process. There is a lot of regulatory agencies that will go through the data, and advisory committee, regulatory review team. There are several groups which will walk through the all the data collected over the 10 years, and they will go through this information, and finally the drug approval process takes place. So as you can see that, like I mentioned in my initial slides, the drug discovery and development process is a very tedious, long process. This entire process I just walked you through takes almost 10 years to complete, and over $1 billion expenditure happens during this process. And even after the approval, also some drugs can be withdrawn because of the side effects reported in the market, and that can also happen. So it is very high risk, high risk environment. I would say just like the life science students investment process. Like we will be spending almost a very long career in our life, right? So we'll be taking to for life science students it takes a long time. It's high risk, but if you get through, you are saving lives of patients, people. So that's the one big thing. Finally, it helps the people to kind of relieve of their problems. So just I can take the analogy like that. So what we've seen so far is drug discovery process, and just as I mentioned, it is not just the single discipline. One person can do everything. It's not like that. It's convergence of various disciplines, and I would say that from time to time, I always say students with biochemistry background are kind of lucky enough. They can kind of understand all this process. In fact, like even chemistry also, we can be like you will be studying in allied subjects, I guess. So you would know the proper, complete information can be easily digested by biochemistry students. And all these fancy names, everything, if you can just if you go through deeply, it does from the biochemistry part only. So I will say that so when you go for in the future, when you go for a career opportunity, and somebody would say that, do you know information about anything about metabolism? Of course, right? Who would not know? Biochemistry we would know the metabolism, we know the enzymology, we know the technology. So don't forget all your basics, all your kind of microelement equation to all the biomolecule protein structure, just your essential, non-essential amino acids. Those informations are really critical for you to go through, understand, and please remember them all throughout your lifetime. So that will be definitely helpful for you. So now I'll move on to the what is new in the current field of drug discovery. So there's a new like we have a multi-omics approach which is currently in practice. I hope majority of you will know that there is genomics, there's proteomics, there's transcriptomics and metabolomics and microbiome. All these things are recently in the past decade has been developed, and it has been used in medical in the pharmaceutical field for the drug discovery development process. So to take a example here, what I can show you is so you can see that how the genome approach is like. So we know that certain particular gene has been involved in the it is involved in some kind of disease process. It has been identified by the genome-wide association study. Example here it is for example locus name is PTO or IRX3, IRX5 genes. And there is a epigenetic process involved, which is DNA methylation or epigenomics process, which has been guiding some transcription factor, which has been guiding this gene expression, and that is evident from the transcriptomics approach. In the normal condition, they are not expressing, whereas in the disease condition, they are expressing in a very high concentration. That's what we're seeing it here. And so all this thing put together, there's a kind of a genomic, there's some mutation which is causing the particular transcription factor to work aberrantly, and that causes the gene expression differentiation, the gene expression changes. So this all can be identified using a multi-omics approach, and there's a functional. Then after this, there will be functional validations in a cell culture method as well as in the animal model, and finally we will find the relevant target identified in the overall process. Will be identified in the relevant process. This is just a short nutshell of what is the application of multi-omics approach in the drug discovery process. Like I mentioned, I've been working through the multi-omics approach for quite some time, and I'll share my thoughts on this entire approach in the next following slides. So that's our one of the way during the introduction process, I've been told what and all I've went through. So for example, so during my I did my BSc, MSc and all, that is fine, right? So how the biochemistry shaped the thing, right? So this is during my PhD, the topic of my research was glycobiology of mycobacterial infection, and we used a multiple omics and drug discovery approach to understand to identify a novel drug target for the mycobacterium tuberculosis. How we went through that is interesting. It is we used a technique called glycocatch method, which is just you would have already studied in your MSc or BSc, simple affinity chromatography. What we did was there is a lectins. These are proteins which will bind to the specific sugars, right? So we immobilize the lectins into the, example here, carn A lectin, into the agarose bead, and we pass through the microbacterium cell wall through that. And once we run through the cell wall, then this slide, you can see that this is part of my PhD work, and the concept in my PhD work was glycobiology of microbacterium infection, and this is a typical biochemistry process we did. So you can see in the leftmost corner, there's a technique called glycocatch we implemented, which is like you would have already studied in your MSCs, right? So it is affinity chromatography basically. What we did was we immobilized the lectins. Lectins are the kind of proteins which will specifically bind to specific sugar molecules in a given mixture. So we used the lectins. In this example, I used a carn A, which is a mannose binding lectin. We immobilized the lectins into the column, and then we passed through the Mycobacterium tuberculosis. What is that? Like Mycobacterium tuberculosis cell wall into that, and whatever bound to the column is basically the mannosilated glycoproteins from Mycobacterium tuberculosis. That's what is that what, right? So after we elute the material using the affinity principle, we use the mannose to elute the glycoproteins out, and then so on the right hand side you can see there's a SDS page image, which is like these are the different glycoproteins eluted out from the Mycobacterium tuberculosis cell wall. Then I went through the process of then I did the mass spectrometry analysis and identified a protein called RB3242C. When I went back and checked the functionality of this protein, we identified this protein was annotated as no hypothetical protein. So that means the function of this protein is unknown. So for that, to identify the function, to potentially understand the function, I've gone through a lot of bioinformatics analysis, simple multiple sequence alignment to the phylogenetic analysis, and then protein structure modeling. All this information led to the discovery of this protein, and it is now like we understood this protein as a phosphoribosyl transferase present in the surface of the Mycobacterium tuberculosis cell wall. Then now, so this is the target identification. Then and so now we need to validate the target, whether the target is functionally important to the mycobacterium or not. So that question we need to understand, right? So in the right hand corner you can see that I did the analysis. So I just cloned this particular Mycobacterium tuberculosis gene into non-pathogenic Mycobacterium smegmatis organism, and found out that this particular after transformed the Mycobacterium smegmatis with Mycobacterium tuberculosis protein, it becomes slightly virulent and it could infect the host macrophages. This and all we tested in different organisms like cell culture as well as in zebrafish mouse model. This particular work has been published in Journal of Biological Chemistry, which is like one of the oldest journal for the biochemistry, for the biochemistry part. So this is one of the good moment. Like we all use the biochemistry understanding to find a particular drug target. Next slide. Hello. Next slide. Yeah, you can click 1, 2, like 1. If you just click the 1, it will automatically go through. Hello. Please click 1. That. Yeah. So this is like so just to we need to understand. So this is just an explanation for the epigenetic mechanisms. When you want to understand what is driving the gene expression, there are like several factors, and gene expression ultimately leading to the heritability of a given trait or something. What is happening is you have like previously it was just understood like only the gene is present in DNA and it expresses. But the current understanding is there are a lot of other factors apart from just nucleotide sequences on the DNA are also involved in the culminating into the gene expression process. Those involves DNA methylation. This one of the prevalent epigenetic mark on DNA that decides whether the particular region needs to be transcribed or not. And this is followed by there are a lot of microRNA and small nuclear RNAs which are also playing the pivotal role in the regulation of gene expression. These microRNAs are basically, if they were found, they will just go and destroy the transcript so that the gene expression will not be happening. So you would wonder, right, why? So all the cells in our body have the same blueprint of DNA, but why certain tissues express certain protein? Why certain tissues express something else? And why in the during the cancer process there is something else is expressed? And so all these questions culminated. All this question is answered by the epigenetic mechanism. So just I mentioned, there's a DNA methylation, there's a microRNA, long nuclear RNA, as well as there is a histone code, there's a nucleosome assembly, which is happening in the chromatin fibers when the it is bound by the different histone proteins. They are just bead. They are just wound around the compressed into the beads kind of structure by the histone proteins, and then that all leads to the process of gene expression, and that is the main reason for the heritability. So it has been, there is a saying in the epigenetic world that like what your grandmother ate actually reason for your current gene expression. So something like that. Same way, what you are going to eat is going to make the in the future generations. So like going into the junk foods and all these things will lead to more and more apparent gene expression in the future generations. So this has been well documented and this has been well studied in the epigenetic process. Next slide. I'm sorry, actually, I tried to do a lot of transitions and effects, but unfortunately, my presentation could not work. Anyway, so here I gave you two examples here. One from my experience of epigenetics. First, in the top part is the one we published with one of the another prestigious journal called PNAS. So in this, what we found was how the environmental agents, like say nickel, say cadmium. Cadmium is found in cigarette smoke. Nickel is found in the exhaust of petroleum products, like smoke from the vehicles. Cadmium is found in the lead in the battery. So all these are found prevalently in the nature, and we have been exposed to always. How this exposure is leading to a epigenetic modification in the system, and that particularly that ultimately lead to the cancer phenotype, say lung cancer, say asthma, different inflammatory disorders. So ultimately, so this is like one of the first ever study which shows that the environmental carcinogens or environmental agents can potentially impact the histone modification in the cells, and that might lead to the expression of different proteins, different genes and proteins, and that ultimately lead to the cancer process. So this is one of the pivotal study which happened, which is first ever study to report this. In the bottom, we have a molecular carcinogenesis. This again. So okay, yeah, the environmental agents are causing the harmful the changes gene expression by epigenetic mechanism, but how they are changing it. So what we found in this paper was, when we introduced the nickel for 6 months into the cell culture in the lung cells, then we remove the 6 weeks. After that, we remove the cells for they just grew the same cells for another 4 weeks without nickel. And what happened is there is a certain set of genes which are persistently affected. That means even after the removal of the particular carcinogenic factor, they still started expressing the same genes
which are kind of causing the carcinogenic process. So this is one such example that, like, even if the exposure is brief and short term, but they can have a long lasting impact. So you can correlate this with the process of smoking, or correlate with the children in the womb. They are getting affected with exposed to certain things, and in the future they can develop a disease, right? So you can, so this can be the one of the you can correlate that way. So it is like how the memory of the gene expression is persisted inside the cells even after the removal of a particular chemical from the environment. And this can be, this process is mediated by epigenetic mechanism. So this is one such, again, it's interesting and important to know that how the long-term effect, how the effect of other various chemicals can cause in our body. So this is one, this is very important to understand, right? Yeah. Please go to the next slide. Just click next. Or you can click the 3, 4, 3. Yeah. So after after I did my PhD and postdoc experience, then I moved into the industry, where my first job is with Vantage Research. It is a quantitative system pharmacology company. So in this company, what we did was we started building the mathematical models for different disease mechanisms, and we started asking, because like I just mentioned in the initial slides, what I mentioned was the drug discovery process is very tedious. Lot of permutation combinations cannot be done in one go because it involves human study, right? So, but what we can do with the mathematical models is the same different concepts can be different experimental like permutation combinations can be studied using the mathematical models. So that will solve the financial problem as well as the time. So you can try different permutation combination using the mathematical models, and then whatever is making sense, you can go and test it in the relevant subjects. So to facilitate that, there's a concept called quantitative system pharmacology. That's what we have been doing. So here it involves the models using the ordinary differential equation. These are mathematical models. I don't want to bore you with a lot of details here. Here, just what you have to know is there is a mathematical process which is helping to ask different what if questions. What if I do this combination with another combination? So we know the efficacy of one combination and separate different combination. But if you combine them, what will be the combined efficacy? You can ask that question using mathematical models. You can generate a virtual patient, virtual cohort, and all this thing you can do with the quantitative system pharmacology process. Next slide, please. So one important thing is right now FDA, Food and Drug Administrative Industry of USA, is mandating the quantitative system pharmacology model to be implemented in the drug discovery process. Yeah. This overall workflow was of the QSP modeling process. It is a kind of iterative and it's a circular process, kind of. It's iterative, means it like multiple steps can be repeated several times, and the tools involved is kind of mostly systems biology tools like MATLAB, SymBiology, and PhysioLab tools. Here what we are doing is combining the data from different sources, from multi-omics approach to multiple cell line information. All the information has been combined into one particular model and derive the values we write the equations for the mathematical, like for the interactions, just like what you do for the enzyme kinetics assays, right? So product versus so two reactants gives rise to product. There is a forward reaction, there's a reverse reaction. That kind of information we try to collect and input that information into the model. Next slide. Yeah, so this is one example. Again, the personal experience. I have created immuno-oncology mathematical model comprising of different parameters. Like you can see it in the right-hand corner. There is a that model. No, what are all the different in the tumor microenvironment? What are all the different immune cells involved? How they are impaired by the cancer phenotype, and how the different drugs can effectively work on regulating those things. So, despite the fact that these drugs are efficacious, we are still yet to know that their actual mechanism, and there is a lot of heterogeneity. So it is working only with just some percentage of the population, not with everything. So, if you want to test that in a global level, it takes a lot of time and effort, right? We can do all this information, all this thing into a mathematical model and test it in the in silico method. So, and based on the outcome, we can say that we can rank the different options, and whichever is best in the cost effective or best for the efficacy versus toxicity measures, we can measure those things, and based on that, we can provide the suggestions to the different pharma companies to carry forward with that specific test, not every test. So this particular work has been with Merck, which is one of the leaders in the IO market, immuno-oncology market. It is like they have a drug called Keytruda, which is like which sells almost $5 billion a year. So we kind of help them to get up to speed with a lot of decision making, and we kind of saved a lot of millions of dollars for them during this project. So just as I mentioned, the how the world is going through the thing. One thing is now we are into the personalized genomics era. So what is happening is that not single drug is not going to fit for all. It is going to take a lot of individualized or personalized decision making to give the patient a specific therapy. It is not no longer we can just give one pill which can solve every problem. It is not going to be happening like that, because we now started understanding the concept like individuals are very different, and there is a lot of heterogeneity involved with respect to the drug metabolism. People are having different mutations, different gene expression in their body, and that is causing them to respond to certain drugs, not respond to certain drugs. All this decision making has been happening currently with respect to the drug discovery process. Now the people are started working on the gene therapy as well as the individualized medicine based on the genome information. Once you sequence the genome, you can know what kind of therapy they will be responding to, especially for cancer, whether they will respond to if there is a mutation in certain genes, certain proteins, then that particular target is not there in the patient. And what is the use of giving them that particular drug which is affecting that target, right? So we don't need that to happen, and so it will add more and more toxicity to the patient only if you just give the drug which they are not going to respond to it. So, but to that end, there is a boom in the next generation sequencing and molecular diagnosis, and so it is estimated that the molecular diagnostics and sequencing is going to be the next big thing in the world, and it is going to be in a big market for it. So that's why it's just I moved to the personalized genomics with the PerkinElmer Genomics in Chennai. So yeah, next slide, please. Yeah, right now my profile is I've been doing the like genome variant interpretation. I'm an India lead for the team. I'm handling the team of 10 PhDs along with me. in my team, and then we all work for the understanding various disease variants, and we are trying to create a clinical reports for them. That's my profile right now, and I also have been like involved in several machine learning kind of like machine learning drug discovery process kind of thing. Like I create a data sets for the new drug discovery process. So I've been working with that end as well. So this is the overall like how the whatever the current technologies applied in the drug discovery process with respect to personal my experience. Right? Yeah, we'll go to the next slide. Yeah. So my fellow students, like what I can tell you is for a career opportunities, there are a lot of options available for us right now. There is always academic research, which I just mentioned. I my whatever I did, and also I was now involved in the industrial research as well. Right, there are several disciplines which you need to you need to be you need to understand before you are venturing out to the job market because like that's what being lacking these days. Like we are not aware of the various opportunities or various terminologies used in the market, and we just keep on looking for certain things and which are not available, and we fed up with that. So I would say that there is a concept called there are different concepts called pharmacovigilance, which is like it is just as I mentioned in my one of the initial slides, there's a phase 4 clinical trial, right? So once the drug has been approved and it is into the market, we have to still collect a lot of information from the different patients, different clinics. All the information needs to be put out in one place in the form of database, and based on that, you can either repurpose the drug for a different purposes, or you can withdraw the drug because of its lot of toxicity. How we will know that information, right? So someone needs to collect that information, and someone needs to put them in a database, and that's how we will be able to get the information, right? So that is one of the nice career options for a biochemistry student. I'm not going to talk about anything like that involves laboratory techniques. Just I'm going to talk about the different options for you in a digital health, like our professor Srinivasan sir mentioned in the beginning, right? So so there are like intellectual property rights, IPR. So there are a lot of legal process outsourcing companies. Lawyers cannot understand the science process, right? So only the science students can understand the science process. So you would fit in into that profile of facilitating them into the intellectual property right in the departments. It can be found in the academics as well as in the academic lab as well as the in the industries. This IPR cell will be there. So in the every year or once in 2 years, there is an exam called patent agent exam. It will happen in you have a IPR cell in Gindi, right? They conduct this patent agent exam every year. So if you qualify for that, you will be called qualified patent agent. And those with the masters and patent agent exam qualification, they can do well into the career. They can just do a lot of they can be into the IP cells of several companies. Several LPOs can be there, right? Legal process outsourcing company, you can get placed into that, right? And of course, there's one more opportunity which is easily not easily, but it is available for life science candidates or regulatory affairs. There are specific courses one need to do after finishing MSCs, but these are really worth doing it, actually. And like that, there's if you are involved in biostatistics, you understand, you do a lot of the small R coding, SAS programming, all SPSO programming. If you can understand the statistical process of experiments and other things, there's a huge opportunity available for you in biostatistics. As same goes with medical writing, medical coding, medical billing. So these are the different opportunities. These are post uh, drug discovery, but these are all the different opportunities available for you in Chennai. And I have mentioned few companies, kind of AstraZeneca put up office in Ramanujam IT company, IT corridor in the Tiruvallur, and Pfizer is there. They even recently they called for the regulatory affairs specialist. So these are all like you have to read, you have to understand different courses. Then based on that, you can apply to them, and definitely you will get through that, right? So this Accenture, TCS, CTS, and all, they have all this medical writing, medical billing, medical coding opportunities. I've known some of our my batchmates are also working in different companies like that. Medical insurance. So only people with the knowledge of clinical understanding, clinical terminology understanding can work in this company, in this profiles. So obviously we all will be fitting into that, right? So definitely we should try into that. Of course, there's a Tyson Biopark where in which my company placed, and yeah, you can just try different companies in that places. Yeah, what I'm trying to walk you through is not just academic research, not that just laboratory research is the important here. You have to. There's a lot of other career opportunities which are waiting to be explored. Next slide. We can skip this slide. It's just all the different opportunities. You can skip this slide. Yeah. Next slide. Yeah. So you would definitely. I can hear you guys are asking where I will find the job in. All the information you just mentioned. For you would have already done this, but just I wanted to just I also did this. During all these years, I just explored and found this information. But these are like for opportunities like Indian jobs. You can just go through Biotechnica, Health Biotech, BioClues. There's a India Bioscience from NCBS. There you can find the information about the jobs, events, conferences. All this information can be found in the NCBS, that India Bioscience website. And for PhD, postdoc, or academic opportunity or industrial opportunity, you can find it through Nature Jobs, Science Career, find a PhD, find a masters.com, and there's a LinkedIn, which can be, it's just like a social media for the working professionals. So just for your information, I found my postdoc opportunity using this naturejobs.com only. So it was posted in that, and then I applied, and I got through that US opportunity after my PhD. Yeah, next slide. Next slide, please. Yeah. So, what is the mantra for success? The key to success is continuous learning. Just your colleague mentioned, reported in my introduction, I still do a lot of certification courses, and I never stopped reading. I never stopped understanding anything. So I stopped. So it always reading, always getting to know the new latest discovery is the key for your success, and you can do it for free as well. There is a NPTEL from Ministry of Human Resource Development. This is like all the IITs and IISCs have put together a lot of courses, free courses from IIT professors. You can take it online, and if you want, you can just write the exam by paying 1,000 rupees, and then you can get the certification for that process. So that will make you someone with the knowledge of that particular discipline, right? So you please use that information, use the opportunity whenever there's a NPTEL. There is so recently this not only that your hard core skills, you should also should have a soft skills like recently TCS. If you have followed the newspapers, TCS recently put up this learning opportunity for which all the students can learn about the communication skills, all the all the opportunity, everything. You can just do 15 days course on this platforms. So that would be really helpful for you. And additionally, for international courses, you can a courses and degree, you can go through Coursera, EDX, Udemy. All these things are for international degrees and courses. I'm still doing a lot of courses in these platforms, and they are really useful for me. And one thing, just I wanted to shout out for the students. This is nothing to have anything personal, but just for the students from the government schools. For your information, I also from the government school. I did my schooling till my 10th standard Tamil medium only, and then later on I started getting like all this English exposure and everything. So just never never just worry about anything. Always try and always be successful, right? So always try to be continuously learn everything, and you will be always be successful. Don't be shy of anything. Yeah. Last slide. Next slide. I'll end you with my profile picture, where in which I received a convocation certificate with my DGVC, and so I was one of the university rank holders. So I got the opportunity to appear in the convocation. Thank you very much. Thank you for listening.
Thank you, sir, for this wonderful and interesting session. Students, now it's time for queries. Kindly ask your questions to the speaker. Those who are attending in YouTube, post your questions in chat box. Thank you.
Good afternoon, sir. Is there any process to learn about quantitative systems pharmacology?
Of course, in the NPTEL, just I mentioned. So in the NPTEL courses, there is a course on biomodeling, and that also covers the quantitative system pharmacology.
Okay, sir. Thank you.
Sir, I just wanted to know if your major work was related to bioinformatics or cell culture work, molecular expression studies.
It is the combination of both. So I start with the bioinformatics work, and then I proceed with the molecular analysis with the experiments.
Okay.
So that's how I
Okay.
proceed. Yes.
Yes, sir. And I can understand that you have traveled a very long path in clinical trial studies. And it's a very basic doubt that I've always had, sir, relating to
Yeah.
the drug solubility. So,
Mhm.
how do you deal with drugs which are water soluble and the drugs that are soluble in organic solvents? Because while targeting, I guess it makes a very great impact, right?
Of course, yeah. So the sense um, to be honest with you, I'm not into that expertise. I don't have
Okay.
that expertise to understand that the solubility part. But what I can tell you is right now there is a lot of formulation options available. You can you can formulate your drug with different biocapsules and different targeted capsule. Even right now what happens is one of the cancer drug from Birkin, what is it, like Bristol Myers Squibb, what happens is it is got the immune targeted, so that means it is covered with a particular antibody which will directly go and bind with the cancer cell and then then release the load into that cell only. So it will not release the load in any other cytotoxic material into that cancer cell only. So they
Okay.
just target the particular organ or particular tissue. So for terminal diseases, they directly inject the molecule into the cancer tissue. So
Okay.
that's one of the recent development also.
Okay, sir. Excellent, excellent. And sir, I would really like to appreciate the way you have put your slides and you have really traveled a very, very, very long path. Congratulations for your efforts, sir. Thank you
Thank
very
you very
much
much.
for your excellent presentation.
Thank you very much.
Thank you, sir, for your lucid answers.
Gratitude is the most exquisite form of courtesy. I invite Secretary of Neuberg Group, Mr. S. Prasanna Kumar, of third year BSc Biochemistry, to propose vote of thanks.
It's more addressed to giving thanks. I consider it a great opportunity and a privilege to propose a vote of thanks to all members who made it memorable and a successful event. On behalf of our Bharat Govardhan Das Vaishnav College, I would like to thank our beloved secretary, principal, and head of the department who took initiative to give us this wonderful session. I would also like to thank our speaker, Dr. Lakshmanan Jagannathan, for spending his valuable time with us, for enlightening and benefiting us with his thought-provoking address. My heartfelt thanks to all our faculties who always been there to guide us and support us. I would also like to thank our technical support team, MC team, and other members of the group who worked behind the scenes to make this event a grand success. Last but not the least, I thank all my fellow students and friends for participating in this webinar on drug discovery and development, current technologies, and career opportunities. Thank you all.